so let's start and let's try to dissection it all together. Try to conquer the brain, but not with mice, but with fish. And that's because I have been working on these nice animals here, which are called zebrafish, and they are small and cute. And you could ask yourself, why should we work and use these animals? But because they are very similar to us. Doesn't really look like so, maybe, and I do agree. But if we go early enough, at the very beginning of our development, we can see that actually many organisms just look the same. Fish, humans, mice, and then, well, we get a bit different. But if we go early enough, we can have enough similarities to take one fish as a model for a baby. And not only at the macroscopic level, but also at the cellular level, I've been working on microglia, which are blood-borne, and indeed the blood cells of the two species are quite similar. So it's scientifically sound enough to be used. However, it's not the only reason why we use fish, but we use them because they are cheap. You can just host a lot of fish in quite a small space. They are still happy, they move around, it's very nice fluid of water, and we can just collect them and put them to grow. And indeed, they are relatively fast. I think many of you are used to work with other models like mice, but I've done a couple of experiments and it's a bit different. You have to plan much more in advance. They do grow relatively faster than other animals, and they are beautiful to watch it. And I would like to just share it with you. This video will show the first 24 hours of development of a fish. And inside you can see 4 eggs. They are inside their shell, which is called chorion. And you can actually see that you have a yolk. And you also have one single cell here. This is the zygote. Basically, all the organisms start from one only single cell, and then they start to divide. And these cells just get quite in shape. And that's exactly what happens here. They start dividing into, maybe. Yes, they do. They start dividing in 2, and then in 4, and then in 8, and then in 16, and then in 32, and then in 64, and then in 128, and then they become a lot, and then they start moving. They start moving, they move along the yolk, they invaginate, they pass around, and then here they are. Here the first part of the back is formed, and here will be formed the head, what will become the head. Here it will be formed what will become the tail. You can start to see the first primordium of the eye. There's retracts. At a certain point, they will start developing muscles, and here they twitch, and they start moving around. And that's only in 24 hours. And for me, it's incredible, and it's nice, and it's cute to look every time. So how do we get these eggs? Well, I am Italian, so we are practicing the art of bunga bunga, and we basically just take 2 of the fish, put them nicely together, let them get know each other over one night, and then magically, when the light turns up in the morning, they start doing what they do. And the female lay eggs, the male leave the sperms around, and then we put a nice net, because this nice caviar here is quite delicious also for them. So without the net, they will just go and eat their own kids. We managed to take them out, and we just collect our eggs. And so what can we do with the eggs? Well, first of all, 1 cage for us is not enough. We want tons of them. And tons means really tons. And in the last 5 years, it took me quite some time to put them up 1 female, put them up 1 male, and then collect their own eggs. And after this, we nicely edit their DNA, we cut, paste, and put inside other interesting genes. For example, this is not a gene, this is a protein. What protein it is?
GFP.
GFP! That's cool! It's a gene fluorescent protein. It's our nice monster. It's been discovered, or like these 3 guys got the Nobel Prize for it. And these guys in particular, Roger Chen, just developed a whole bunch of nice color with which we can produce such stunning fish. In this case, they have not been produced by our lab, but we have some fish that have been labeled green fluorescence in the vessels, and we have neurons in red, and a particular protein, which is the fibrillar protein produced in Alzheimer, that is highlighted in blue. And how do we get from the DNA or from the protein to this beautiful fish? By painstakingly injecting each and single one of them. So you basically start in the morning, you let the fish do what they do, and then you get the eggs, and then you sit at this nice microscope, which is paired with a microinjector, which is a fancy stuff to say a small glass needle that you painstakingly remove by hand. And indeed, here are our friends, the single eggs, and you go inside the single cell, and you put a little bit of DNA, and you hope that it gets inside. So this is more or less the model. This is what I have been using for years, and the aim was to study these guys here. Who are these guys? I know the Italian knows them. What are they?
Yeah, white blood cell.
Exactly, that's white blood cells. We call them macrophages. There are lots of white blood cells. I see that the audience is prepared. Did you select them, or do they have a test at the entrance? That's awesome. Yes, so these white blood cells that are called macrophages, they exist in different states inside our organism. They basically go around the vessels, and this is the circulating population, but they also go outside, and like policemen, they patrol single parts of the body, single organs. And inside each organs, we have our specific population of macrophages, and they get super strange names. They are called Kupfer cells, they are called Langerhans cells, and in the brain, they are called microglia, because they are small, and they are glia, which means not neuron, which is quite fun. I was trying to understand during my PhD how these cells get out from the blood and how they populate these organs, and if at all they need to pass through the blood or they can take other routes. And indeed, macrophages are tons. There are small dots here that are red and that they move around, and I'm proud of this. Scientifically, it doesn't mean anything, but it's beautiful, and I made the line, and so I can show it now, and I'm quite happy about that. So how do microglia look like? They look like stars. This picture is quite old. It was done already in 1919 by Pio Deri Ortega, which perfected a technique developed by an Italian and by Camillo Golgi, which you all know, by the Golgi. But Camillo Golgi also developed the technique to stain cells inside the central nervous system. And when they got the number together, Golgi behaved really like an asshole. He just trashed all the theories of the Deri Ortega. But it turns out that Golgi was wrong, so it's not nice for us Italians, sorry. But it's good because he really pinpointed it. But if you look at like these, they seem quite boring stuff. Beautiful but boring. But in reality, they move a lot. They move around, they branch, and they go around and seek for dead neurons. And what they do, they just pick up these dead neurons, they eat them, and they basically clean up the whole brain. And indeed, if we look at the brain from the top, this is the brain of a fish, and you can see the height here. If you look inside, we can see that there are around 25 cells when the fish is properly developed, which just move around and scan all the parenchyma. And what they do, they look for damages. And if there is no damage, well, that's not a problem, because they clean it up. Anyway, they have always something to do. And if there is no main damage, there are scientists that make them. And that's what we do. We shoot with a laser inside the head and see what these microglia do. And what they do is they dance, they move. They branch towards the damage, and they just quickly go towards it and try to repair it. Which they fail because we shoot a big laser inside a small head of a small embryo guy. But they try at least, and that's it. How do they get there? I said that they dance, but actually they surf. They basically use waves of a small transmitter, which is calcium, and you can actually look it here. When we make this damage inside, there is calcium just spreading all over the brain, and the microglia feel it and just moves towards. So I hope that I was clear enough. and I will try to sum it up, so join me with this song. Which way out there will it take me? Through the matrix, open always in the gates. And ask the question, but we can see them moving. There is no time you run. There is all their holes on how to play. They look as and good, and you feel all the natural way. This is my past. This is what I did in these 5 years. At the end of the evening, there will be a small talk that I kindly ask Basia if I could, and I would like to ask you to think of a topic that you would want me to speak about. It's a science talk, so just get the topic, think about it, and I will ask you later.
Does anybody have any questions for Federico? So I have to say that it was a great presentation. I am very, very far from neuroscience, but I could understand everything, and I hope that everybody did. So if no one has any questions... Oh, I have a question!
Get that girl a microphone back there.
Ciao Federico, grazie. My question is actually, how do you think the microglia move? I mean, they sense calcium.
Yeah.
Do they move along the extracellular matrix, or do they move along some other structures in the brain?
So the brain is packed with neurons, and there is an area where you have made cells body, and they squeeze through it. And you can actually see, because they really branch around, they don't have much space to move the body. And at a certain point, if they really sense that they have to move in one direction, they extend the branches and then drag along the body. But there is no track.
No track, okay.
Or at least not that we know.
Thank you.